Okay, so we've had long and good time with biochemistry all the way into cells. That is subcellular. We've done all sorts of cool things kind of within the membranes. Most specifically and most recently, aerobic respiration and photosynthesis. Now it's time for us to turn our attention to whole cells. And the first thing we have to think about with whole cells is the fundamental process of cell division. So this brings us to chapter 12 in the text. And we're going to basically be looking at what's called cellular reproduction. Now, we're going to go through this process, and most of the time we're going to be spending our time focusing on animal cells. We're animals, it makes sense. There's slight variations as we look into plant cells and fungal cells and single-celled organisms, but we're going to stay right here with animal cells. And so if we draw a typical cartoon of an animal cell, we've got our phospholipid bilayer, you're used to that. Then we have this nuclear envelope, and of course I'm drawing double membranes here. I'm not going to throw any organelles in at all. All I want to do is kind of indicate that when you look at a cell under the microscope, you can see that the nucleus is actually darker than the rest of the cell contents. Of course, with the exception of maybe a plant cell whose cytoplasm is completely filled with chloroplast. But in an animal cell like this, this is what they look like. And that sets us up. What's inside of this of the nucleus is a lot of big-time biochemistry. So inside the nucleus at a cell in a typical state would be things we call chromosomes. And the chromosomes at this point, let's define chromosomes. So these are long specific molecules of DNA. Of course, they're macromolecules. Macromolecules of DNA. We know what DNA is. We've already defined it, but I'll go ahead and refresh your memory. Deoxyribonucleic acid. So there we have the D, there the N, then the A. Of course, we talked about that. They're made of nucleotides. You remember what those are? That's a sugar, a phosphate, and a nitrogenous base. Further, you would remember that there are purine bases and pyrimidine bases, and that in DNA, the four bases are adenine, guanine, cytosine, and thymine. We've got a lot more to say about DNA later, so we won't spend too much time. All right, so those are chromosomes, and the deal is that these chromosomes have genes, and genes are specific sequences, base sequences that code for specific amino acid, and I'll abbreviate that, base, amino acid sequences. So that's our kind of general look. And of course these amino acid sequences, we would know in the big picture that these things are later going to be kind of put together in, folded, twisted, turned into proteins. And proteins are molecules of life. You remember that we've looked at structural proteins. You probably remember that usually we identify structural proteins with an IN ending in their kind of common name. Think hemoglobin, actin, myosin, and then of course enzymes, and you know how we put those names together as well. Enzymes always have ASE endings. So proteins are the building blocks of life. Here's the whole story. Now we focus right back here to the cell, because what we want to do is kind of set the stage for a radical process that is going to take place here. Before we go very far though, we have lots of chromosome lingo that we need to get under our belts. the kinds of things, I use the word specific over here. Specific is going to be a big, big part of this. So I think I'm going to need more board to pull this off. So I guess let's just get this thing done. So organisms, this is very important, big star here. Organisms have specific numbers, I'm abbreviating that, specific numbers of types of chromosomes. Okay. This might start off confusing, but it won't be for very long. For instance, in Homo, we have 23 types. What we use for identifying in code what these numbers are, we call the number of types the N number. So this whole thing, this specific number, this is called the N number. N. So in Homo, N is equal to 23. We have 23 different types of chromosomes. As a matter of fact, I'm going to take you through a little tour of what's called a human karyotype on a PowerPoint slide a little bit later, so we can see all of the chromosomes of a typical human cell kind of laid out in a photoplate. That's what a karyotype is. So Homo, that's our genus. We have 23 types of chromosomes. Some of you might even further know that we have a number system, and therefore, so within the chromosome systems we have a number system, and a general rule for the number system is that the lowest number is the largest, and then down to the smallest number of chromosomes will be the smallest. So in humans, we have chromosomes 1 through 22. So number 1 through number 22, and this is the largest, this is the smallest in the numbered ones. And then we also have, not really a 23, we have the sex chromosomes. We call these chromosomes here the somatic chromosomes, and we call then the sex chromosomes, which we have subtypes, X and Y. And interestingly, X is larger and Y is smaller. And there's an important statement to be made here in the general concept of biological feminism. Females have we'll find out, that the sex determination system for us is XX and XY. XX being females, XY being males. This is a smaller chromosome than this chromosome. So females are superior to males in Homo sapiens because they've got more DNA. Simple as that. Okay, our next one. I told you that we were going to take a look at the human karyotype. And the human karyotype is a grand example of us being able to use our formula for ploidy, which was X times N equals total. So this is a human karyotype. Let me first tell you how this was obtained. This is obviously a very high quality photomicrograph. In other words, a picture taken through a microscope. But prior to that, a sample had to be prepared. So what happened here was that a pregnant mother had some amniotic fluid drawn, which actually contained fetal cells, because the fetus is this rapidly developing by mitosis and cytokinesis kind of embryonic stage. And it's easy to find cells that are in mitosis in that. So what the karyotype technician does is find a cell, use special techniques to crush the cell. Remember, at this point, the chromosomes are duplicated condensed chromosomes, so they kind of bleed out of the cell all over the slide. And then basically you take a picture and then you use modern digital techniques to lasso a chromosome, bring it into position, rotate it to the right place. Boom, 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 all the way down from number 1, which as I told you before, we use a numbering system for somatic chromosomes where the low numbers are the largest chromosomes and the small chromosomes get the highest numbers. So here we see actually, therefore, each one of these is both of the chromosomes of a diploid kind of fetal cell. And they're putting them right next to each other. If you actually could blow these up and take a look at them, and matter of fact, when you see this, some of them actually have, you can see that what you're looking at is basically that, a real chubby X. There's a constriction at the centromere region, and then a chubby X, each of the chromatids. So that's what these are, and so therefore we've got, for number 1, we've got 1 and 2, number 2, on down the line, and notice that right over here, things get interesting because we have an X and a Y. So naturally, therefore, this fetus that produced this cell, that was sampled from the mother, is a boy. It's a male coming along. Now, XN equals tote. So our X is going to be 2. This is diploid, we have 2 sets. So there are 2 of each. 1, 2, 1, 2, 1, 2. And then our N number, as we know for humans, is 23. And as I told you, 1 through 22 are somatic, they get numbers. And then the sex chromosomes, which are subtypes, X and Y, don't get a number, but that represents our 23rd. So N equals 23. We've got 2 of each. 2 times 23 equals a total of 46. If you count these, you'll end up with 46. Now, one other thing to say about human karyotype that's particularly interesting is that in genetics and reproduction, some things can happen where you end up with a trisomy. Now, we already know somatic is referring to chromosomes of the body, so a trisomy might be referring to having 3 instead of 2. And one of the most famous trisomies is on little chromosome number 21. What they're looking for in amniocentesis is they're screening for a trisomy at the 21 position because that is something that the fetus can survive. Most trisomies are fatal, spontaneous abortion, whatever you want to call it. But a trisomy in this little tiny chromosome here actually has severe, potentially severe, developmental consequences for the the developing fetus all the way into life as an independent human. And a trisomy 21 is a syndrome. We call it Down syndrome. And it produces different degrees of morphological and developmental kind of impairments. All right, so this is what this looks like. And now it's time to break into some really, really cool stuff in relation to this N number. But before I do that, I want to let you know that different species have different N numbers. Now, there's not a general rule that the bigger your N number, the more evolved or the more advanced of a species that you are. Get that out of your head right now. As a matter of fact, the botany world presents some extraordinary N numbers. There are actually trees, there's some palms that have N numbers well into the hundreds. And if you actually tried to look at all of those chromosomes and do the tally, it's called cytogenetics. It's this great 20th century kind of pillar of scientific study. I mean, it still goes on now, but back in that day, it was at its peak. And people would have to be faced with the problem of how to see all of these chromosomes to determine N numbers. And when you do this, you end up with the chromosomes at their visible point. We'll talk about that in a moment. With just a huge mass of whole tiny little chromosomes is basically what the deal is. And so I think one of the concepts that we talk about from the course of the span of organismal diversity is the fragmentation of the genome. So there, basically, the genome is the sum total of the DNA of an organism. And that's all the DNA. So if then you might have an idea that, you know, you've got this, theoretically, you've got one big huge piece of information, one big huge piece of DNA, and then it's actually little smaller fragments of that actually become functional chromosomes. You're going to see that being a small chromosome instead of an incredibly large chromosome is particularly important when it comes time to duplicate the DNA and then duplicate cells. Every cell that every cell in our body came ultimately through the very, very beginning of us being a zygote. One cell, one nucleus, as we will find, two sets of chromosomes. So, lots of stuff to think about here. Get back to this though, the N number is the big deal. The N number is a specific number of types of chromosomes. We have a number system where the biggest chromosome gets the biggest number, the smallest number, and then in some certain cases, especially like us in Homo, we have sex chromosomes with subtypes X and Y. They don't get a number, they get, in our case, X and Y. So that's that. Okay. So let's get aboard, Erasure. All right. So terminology. Terms related to the number of sets of chromosomes in a nucleus. I'm abbreviating nuke. So the number of sets. This whole concept here we refer to as ploidy. Ploidy. And so we have this situation where there's different conditions. We would consider a cell as being haploid, or excuse me, a nucleus as having one set of chromosomes. Then diploid would be two sets of chromosomes. You could actually continue this party and do triploid, which would be three sets of chromosomes, and you can continue that process. At some point, we usually don't call diploid cells polyploid. Poly, though, meaning more than one, so technically it is. Right around here, though, we could say that, oh, okay, these guys are polyploid. And technically, as I mentioned, perhaps a diploid cell is theoretically polyploid, but that's what we have, the number of sets of chromosomes. And then what I want to do is kind of put this all in the perspective of just our basic, fundamental, simple life cycle. We aren't too fancy in the life cycle department, but we know our life cycle. I mean, everybody here knows the life cycle. That's what you're doing. Okay, so more board. Let's take a look at us. Okay, there we are. And then here we are. Okay, that's just a little sketch of the business. So here's the thing. We've got here Whoops, that's the wrong place. We've got here, we've got here. Very important parts. We have here, and we have here. Very important parts. So what I wanted to do is show you that everything that's white is 2N. And, oh, I didn't even give you that code. So I can say everything is diploid. That equals to 2N. I'll tell you about that in a minute. This is haploid. Or at least it's producing haploid. Not the testes themselves aren't haploid, but they're producing it. And then ovaries over here would be haploid as well. So producing haploid. And that's the business. So if we take these haploid cells here in our life cycle and join them with these haploid cells here, we end up making a diploid cell, and this would be what we call a zygote. And so that's one special cell. And what I'll do is I'll put both blue and red in the nucleus so we can see that, hey, yeah, that's beautiful. So we all started off with getting one set of chromosomes from our father, one set of chromosomes from our mother. And then the process here is that depending on, if it goes this way, if it's XX, we're going to go and grow into the female animal. And to do this, we're going from one single cell to radical multicellularity. Over here is the process also of going from having XY condition. Matter of fact, I should do this. Okay. There we go. So here we have an XY situation growing into the male Homo sapiens, and it's these processes here, which actually we should highlight with many, many, many arrows. Each arrow equals, huge numbers of multiplications. This is mitosis, the subject of the chapter, and cytokinesis. I'm going to define both of those things here in a minute, as well as defining some of these numbers. And then this, of course, here is also mitosis and cytokinesis. So there you have it. That is our life cycle. That's our beautiful, beautiful life cycle. I think everybody knows how this happens. There's all sorts of exciting life here. There's behavior involved, and we need to do this. This is sexual reproduction. Now, the question I want you to answer is simple. Can we, Homo sapiens, reproduce asexually? That's the question on the table. And I imagine that you all know the answer immediately. Big fat no. We cannot reproduce asexually. There are some animals and some plants and some other organisms that not only can reproduce asexually, but some of them absolutely rely on it. In my Corona break here, I've been watching lots of British garden shows. And time again, the host of the show will get into his garden and he uses the word cutback. He'll snip. And what he will then do is prepare a little shoot, a little piece of a plant in such a way that he puts that thing into soil and waters it, and that clipping becomes a whole new plant. Can you imagine if you could do that? You could clip off maybe you have to clip off the tip of your pinky finger or the last the last digit, and set that thing in throw it in your closet in a bottle of beef broth or something like that, and check on it, and it grows, and then all of a sudden you've got a new human. It doesn't happen that way. But my purpose for actually kind of stimulating these thoughts is for us to kind of embrace ultimately the power of sexual reproduction. So when we get to chapter 13, so I'm going to get some colors here and say, yeah, this is chapter 12. This is chapter 12. This whole part right here is chapter 13, where we are going to focus on making gametes by a very special process called meiosis. And therefore up here, this of course is mitosis. Chapter 12, where we are now, we're going to be focusing on cells dividing and making genetic equivalents of the cell that went into the process. So a cell cloning process is really what we're looking at, a natural process where that really defines how multicellular organisms grow. Multicellular organisms start with one, in sexual species like us, start with one cell that has two sets of chromosomes. One came from the female, one came from the male, and that thing is going to grow by division into to mega multicellular organisms. All right. Another little highlight of this process that we could throw in, I want you to think of the processes of kind of continuing. You know, most of us are grown. What's the phrase? I'm a grown-ass man. But I'm still dividing. Every single day you are scraping cells off of the insides of your endothelia. Those are the layers of cells that line your tube from mouth to anus. And you're scraping these cells off, and they're passing out of your body, ultimately in bowel movements, right? We have to replace those cells. So we have active division to replace cells constantly in our endothelia. And of course, we're also doing similar things on our outsides. And so even though the peak of mitosis is way behind us, we had mitosis and cytokinesis happening radically from when we were zygotes until we were, you know, something that actually stopped growing. And it was really fast at the beginning. So the point that I'm trying to raise here is that as we venture into the details of mitosis and cytokinesis, which together mean cellular reproduction, we need to not just assume that it's a static phenomenon that's happening at the same rate in an organism all the time. Different tissues are going to have different rates of cell division, and certainly early development with embryonic tissues is the peak of division. So then, with that in our minds, you have to think about, okay, today is a competition. You all have to go out. This, of course, is a theoretical competition. You all have to, when I say start, boom, you all have to run out and get real samples of tissue that we can look at under a microscope that are going to show the highest degree of mitotic division. Because you can see mitotic division in the light microscope. So, what are you going to do? Boom, start. And then, of course, some of you are thinking, oh, okay, I got to go out and find something. Well, don't just cut a chunk out of yourself and put it on a microscope slide, because there's not going to be a lot of division right there. You need to find things that are embryonic. That's the peak of division, and that's when you can see the highest proportion of cells that are engaged in the mitotic process. Okay, so that sets the stage. Got a little bit more work to do on numbers in relation to ploidy, so you can carry that through, and then we are ready to celebrate an extraordinary process. Okay, I told you we're going to deal with numbers in ploidy. So I mentioned that we have ploidy. Ploidy, the n number, which you don't know what this is. Well, you do. I mean, you do know what this is, and what we call the total number of chromosomes. And all of this that I want to talk about is in a nucleus, or for a nucleus, we describe this. So we have a formula, the beautiful formula. And this formula is xn equals tote. So n is the n number. X is equal to the ploidy. And x equals 1 if it's haploid. X equals 2 if it's diploid, and so on and so forth. Okay? And the beauty of this is that all you have to do is multiply these two values to get the total number of chromosomes in a nucleus. So for example, we have a human somatic cell. Somatic cells are cells of the body, so they're not gametes. And therefore, xn equals tote. So we know that human cells are 2, and we know that our n number is 23, so our total number of chromosomes is 46. So in your cells, you've got, in the nuclei in your cells, you have 46 chromosomes, with the exception of gametes that are produced by a very special process called meiosis. So that's human somatic cell. Let's erase this bottom here and do a human gamete. Human gamete would be xn equals tote, and in this case it's haploid, so it would be 1 times 23 equals 23. So gametes have 23 chromosomes. Now, with this formula, this grand formula right here, xn equals tote, with this grand formula you can solve all sorts of different kinds of problems in relation to questions asked about chromosomes in nuclei. So I'm going to give you a few examples, a few things that I'd like you to work out. Okay, so here's kind of some ploidy problems. So I guess that I could say how many chromosomes are in a diploid nucleus with an n number of 5. So that's one to solve. Number 2 is, what is the ploidy of a nucleus with n equals 3 and a total of 9 chromosomes. Okay, number 3, how many sets of chromosomes are in a nucleus with an n number of 8 and a total number of a total of 24 chromosomes. So there's a few problems that you could work. Just made those off the top of my head. How many chromosomes are in a diploid nucleus with n number of 5? So xn equals equals tote. So I've given you the n number. So that is right here. And then I've already said that it's diploid. So that's 2. And 10 is our answer then. So 10 is our number there. That's what we're using the formula straight up. What is the ploidy of a nucleus with n equals 3 and a total of 9? So xn equals tote. And in this particular case, we want to know the ploidy. We have the n, it equals 3. This remains x. And then tote equals 9. So here we actually have to do some basic math. So solving for x, it's 9 divided by 3, therefore x equals 3. So this would therefore be 3n is a numerical representation, triploid is the word for that one. Okay, how many sets of chromosomes are in a nucleus with an n number of 8 and a total of 24? So this is xn equals tote. And here, our total number, I said, was 24. And our n number was 8. And it's the same thing then. We're solving for. Same thing, but different wording. So here it is x, solving for x is 24 divided by 8, which equals 3. And so there are 3 sets. That's what I've asked. Okay, so there's some problems. I want you to think about this. This is one for you to think about, and I'm going to put it on the board. So we'll see what we can do here. Okay. So, for thought. What is the ploidy of a deck? Ploidy and n number of a deck of cards without jokers. So what's the ploidy and n number of a deck of cards without jokers? Think about that and come up with a good answer. I don't know how I'll ask you for the answer, but I'm going to put this one in your head, and I want you to think about it. Maybe I'll give you an opportunity to tell me the answer. Okie doke. So, okay, now it is time for us to look into the cell cycle. We're going to take a bigger snapshot of the cell cycle to identify some of the fundamental steps that occur. And then we're actually going to switch over to the big white paper so we can draw the actual mitosis process. So the cell cycle is, for us then, going to involve 2 broad things. The cell cycle is going to involve interphase, which we will further divide into what's called G1, S, and G2, and then it's going to involve mitosis, and the way we're going to do it is plus cytokinesis. Well, actually, I'll leave that down here. I'll say cytokinesis. We're talking about a cell cycle that actually produces 2 complete cells from 1. So cytokinesis. And mitosis is going to involve the following steps. Prophase. They have one called prometaphase. I guess we could go and take a look at your book. They have prophase, prometaphase, and then strict metaphase, and then anaphase, and then telophase. We're going to draw these glorious, beautiful diagrams. So prophase, prometaphase, metaphase, anaphase, and telophase. And then after that is cytokinesis, which actually is its own thing, which is the division of cytoplasm between two new nuclei. So intermitosis produces daughter nuclei. So I'm going to use the word clone, which implies genetic equivalency. So produces two clone nuclei in our simple way. And then cytokinesis is actually going to be the division. Oh, and by the way, let's put this in. Two clone daughter nuclei. Let's throw daughter nuclei here. That implies that they are the products of mitosis. So this is the division of cytoplasm, membrane, etc. Cytoplasm between daughter nuclei. And that's that. Okay, so we're going to actually have lots of great illustrations to tease out the important elements of this. But the first thing that we want to do is to kind of put it into the concept of a time cycle. So interphase, mitosis, and cytokinesis, let's take a look at this on a time perspective. So if we were to take a look at kind of the face of a clock. This is the face of a clock. So suppose, therefore, that this is like if a cell cycle in its completion took 12 hours, then this is a perfect tool for us. So this would be, kind of the clock face. So what we would see in our deal is the first part running down to about here. This is a phase. The second part running over here. This is a phase. And then there's a little variation here. This is not just like fixed hard times. This is a typical cell cycle. And then up here, we would draw something that we want to really stand out in red. And then right after that, we'll draw something that we'll put in as... Actually, I don't even know if I want to use red. I think I'm going to change to orange. Because red indicates stopping and green indicates going. So in orange here, I'm going to put this right here. And then in another color, to be determined, ooh, purple, that looks good. I'm going to draw something that's right here, right before midnight. So if we had the 12-hour cycle, let's label these parts. This part right here is called G1. This part, right, actually I'm going to write this inside and then we'll label these. So this is G1, this is S, this is G2. So you saw that on the previous board. And then big orange here is M. That's mitosis. And then this one here is C, which is cytokinesis. So this is our process. What we need to do, though, is put some more stuff in here. We need to throw something in about right here and label this as the G1 checkpoint. Then we need to throw in another checkpoint right here before it goes into M. And this one, we're going to call over here the G2 checkpoint. And then, right in the middle of mitosis, here, right here in the middle of mitosis, we're going to have an M checkpoint. So there we are. That is our overall kind of view of the process. So let's identify some of these things. G1 is something that we could basically describe as organelles in the cell. Organelles and ribosomes are duplicated. So the whole cell is preparing for division. G1, that's what's happening. And if this was a 12-hour cycle, this is taken until about 4:30 or 5 p.m. This is 4 or 5 hours in duration on a 12-hour cycle. S is the process that, I guess I'll put this down here. So S is going to be our process of DNA duplication. Very important here. DNA duplication, and in this particular state, I want to say to form duplicated chromosomes. More on that later. In quotations, duplicated chromosomes. Okay, duplicated chromosomes. And then we move over here, we got G2. So G2 is kind of a period of arrestment. There's not a lot going on in G2 other than this checkpoint, which we'll talk about here in a minute. So, one thing that could be said about this, imagine that you're looking at a microscopic kind of monolayer of cells, a tissue of some kind, from an embryo, or from the parts of plants that actually are rapidly dividing and growing, like some kind of a bud or a tip of a root. And you're looking at that layer of cells. And basically, since all of the fireworks starts in about the last hour, that most of the time you would not, most of the time, all these phases in white, the cell would look just like the normal cell. So these phases in white, the phases in white are going to be looking like this. Here's the animal cell, here's the nuclear envelope, and then you would actually have, stuff in there because there's a lot of big molecules in that nucleus that are held captive there, those chromosomes that we talked about. So this is what the cells are going to look like. And then, by the time you get into the metaphase part, the metaphase cells are actually going to look like a lot of different things. As a matter of fact, the mitosis ones. I'll actually show you metaphase since I blurted it out. So, as an example, and here's something well established called a spindle that you can see, and then our chromosomes are all awesome structures that are kind of lined up on the equator of the cell. These spindle fibers need to be smaller. So that's that looks radically different from that. It still has the same membrane, still outer shape, but inside you can't even see the original nucleus, and you can see these chromosomes that are thick and chubby, and they're all held in place by smaller lines that are microtubules. So this is fantastic stuff that sews together lots of our fundamental biology. And then, by the time we get into C, in the case of an animal cell, we're actually dividing the cell into contents and perhaps even forming a new daughter nuclei, and things are starting to happen. So this would be an example of what's happening in C. So by color, most of the time, cells look like this. We see their nuclei. We see that the nuclei are darker stained than the surrounding. And then when we get into metaphase, we see not only metaphase, but prophase, anaphase, and telophase. Actually, telophase is where we really are here, technically speaking. Actually, here's the cytokinesis is the end of that. And so by the time we get to cytokinesis, telophase has already occurred. The daughter nuclei are now kind of in each of their new cells. So these, of course, are going to be daughter cells. And the duplicated chromosomes are kind of uncondensing, getting back into their natural state and beginning to look like this. So I guess that's what the cycle does, is that we go back to there. Each one of those cells would go back to there. That's the cell cycle. Now, we have a little bit of work to do on our checkpoints. So we've got 3 checkpoints. Checkpoints are red lights for traffic. Checkpoints are red lights for traffic. Matter of fact, let's go ahead and throw that there. Here's my red light, boom. And there's my red light. And here's my red light. These checkpoints are awesome. What has to happen to turn the light from red to green is that there has to be kind of a strong cellular signal that, hey, we can move forward. So I want to tell you about the signal that's up here, the M checkpoint signal. This is an example of what happens here. So this thing is in the red light position. This will make sense a lot more once we actually lay everything out, but this will make sense later. When the red lights are on when the duplicated chromosomes, again, I'm naming that later, the duplicated chromosomes, which I'm going to abbreviate, are not attached to spindle microtubules. So this thing called the spindle, and again, we'll be talking about that later. Green light is when duplicated chromosomes are attached to the spindle, on both sides, by the way. So that's an example of a checkpoint. There's work to be done. Things had to happen, and that signal is only going to turn from red to green when the chromosome is attached on both sides to kinetochore, or something we'll talk about briefly in a moment. So these spindle microtubules are attached, and the whole thing is established, because that's the scaffolding with which we're going to be able to actually send duplicated chromosomes to break apart, and then chromatids are going to move to opposite poles. All that language comes soon, very soon. So we have a checkpoint here, we have a checkpoint here, and again, the key is basically to go to green. So to basically advance, to get the green light, requires signaling. Cellular signaling. And it is a little bit, we don't have this comprehensive understanding of signaling kind of coursing through our brains right now, because that was a chapter that I've elected to kind of leave behind. So if you're interested in signaling, that's intense stuff, have a look. This, therefore, again, we would get green light in this checkpoint that requires signaling. So to get the green light requires signals. So there we have a beautiful, beautiful board that establishes in our mind that the cell cycle is a very long and beautiful and dynamic process. Most of the time, even though things are happening in a cell, they look like cells doing business as usual. They've got nuclei that are easy to see, and the nuclei are darker because that represents all of the chromosomes and other big molecules that are kind of held prisoner in the nucleus. And then a lot of our cells are just in a checkpoint. They're in a stop position. It's not time to do anything. And anyway, we continue through, and if we clear these two checkpoints here, when cells are looking like this, we actually enter into mitosis, and the cell radically changes, beautifully changes, especially under the microscope. That's when you really get to celebrate this. And they go through these processes: prophase, metaphase, prometaphase, anaphase, telophase. And then finally we switch over to let's finish the job and make 2 cells, 2 clone cells, 2 equivalent cells from the original cell. We call them daughter cells. Boom, that happens here. And guess what? Each one of those cells then re-enters their G1. Cell cycle, cell cycle. That's what we are. Products of the cell cycle. Okay. Okay, now it is definitely time for us to get into this extraordinary process of mitosis. So here, let's see if this shows up. This will be mitosis and cytokinesis. I gotta check and see if we get this in the camera. Oh, we certainly do. So, with mitosis and cytokinesis, we're gonna actually start just with a series of drawings. So, we're gonna actually come in, this first part really is gonna be pre-mitosis. And of course, we know pre-mitosis is interphase, so let's draw this cell. So here we actually see our stereotypic, kind of nuclear envelope. We do have some interesting things to talk about, however, in this process, because right here I'm drawing a couple of these little structures we call centrosomes. So this is going to be what we call interphase then. And in interphase, we will actually have our chromosomes. I'm going to color code our chromosomes. More on that later. So right now they're all just beautiful and wound in the brilliant location of the nucleus, kind of doing their thing. This is interphase. So you can see that the nuclear envelope is very, very apparent, and the centrosomes are here. Remember that in interphase we had G1, we had S, and we had G2. And it's actually in S that these chromosomes are going to be duplicated. But this is what it looks like. Now we can move into mitosis proper. So kind of put this line here. So now we're actually entering into mitosis. And mitosis is going to start with this process right here. This process we call prophase. And in prophase, we're going to actually see that the nucleus is still pretty tight. You can still see the nucleus, but the changes that are occurring are manifest in the movement of the centrosomes with their spindle microtubules forming here. You still see the nuclear envelope. It looks pretty tight. And you can now see actually these chromosomes appearing. And these chromosomes are X's, like this, and I'll explain that in a minute. So here is a beautiful long one. You can put these wherever you want. This is beautiful. Now, at this point, we need to get some information. Why have I drawn things the way that I've drawn them? Well, in our example, our cell, what we're going to use is 2n, it's a diploid cell therefore, n equals 3, and our chromosomes will be long, medium, and short. So we have long, medium, and short chromosomes. Now remember what this equals. This is what we can do. So we have xn equals tote. And we already know that our x is 2, our n is 3, so therefore 2 times 3 equals 6. 2 times 3 equals 6, and it's actually going to be, I'll grab a finer pen here, this number 6 here. Count them up. So you have 1, 2, 3, 4, 5, 6 kind of twisted x's. The twisted x's are called duplicated chromosomes. more on that later. So that's what happens in prophase. Then we move to our next phase, which is metaphase. And in metaphase, your book introduces something called pro-metaphase, by the way. I'm actually going straight for metaphase. We're going to skip a step. So the book has pro-metaphase here, but we're just going to do metaphase. And by the time we're in metaphase, these centrosomes have completely migrated to opposite poles. And by this time in metaphase, the nucleus is completely fragmented. So, and then our beautiful chubby chromosomes line up. They're chubbier than they were the last time. So that's a medium, this is a short, and then here's a long. Look how chubby they are. And then, let's see, I'll throw in the long maternal chromosome here, so the long red one and the medium one. So there we have that. And now the beauty of this is that we can draw complete and beautiful spindle microtubules to each of these duplicated chromosomes. That's that. So our spindle is complete. I guess we can draw some of these like this to show, that the spindle is complete. That's what metaphase looks like. So chromosomes are lining up on the equator. We'll summarize all this here in a minute. And now we'll move to the next phase, which is called anaphase. So in anaphase, obviously our nucleus is completely gone. We have these lovely centrosomes at the poles. And now what's happening is that these duplicated chromosomes are going to break apart, and these chubby little chromatids that are still condensed are going to migrate to opposite poles. So there's the long one. Notice how they're kind of folding back. They're being poled, positioned. Let's see, that should be pretty big. And then this one's supposed to be our medium one. I'm going to make these even bigger so that we know long and we know medium and we know short. Okay, so there's a medium one coming back like that. Of course, we have to do the same with our blue chromosomes. So there's a big one, there's a big one right there. And then we have a medium one coming here, medium one coming here. And then I have to sneak in the wee little blue one right here. And then let's go ahead and show these spindle microtubules doing their thing. This is great. And so that's what that looks like for us right now. And there's anaphase, and then we move to this next step, which is a division of cytoplasm between the two. This is actually the telophase. In telophase, we're going to see the beginning of the kind of reappearance of our nuclear envelope, and we've got that. It's going like that. So now we want to draw our chromosomes, but we're going to draw them differently. So there's a long, there's a medium, there's a short, there's a medium, there's a short. Those are the blue ones. And then we'll have here a long maternal, a medium maternal, short maternal. So that's beautiful. Wow, that's spectacular. This is what we call telophase. And pointing out an important feature here, this feature would be called the cleavage furrow. I guess what I wanted to do here is get a color on this. So I'm going to color in these structures here all the way, and then I'm going to label what they are. This is more what it looks like in the book. Coloring in these structures. So these things are called centrosomes. And the centrosomes, I think we should also color our spindle microtubules, kind of highlight those. It's going to look very, very much like the figure in the book, but better because we did it. And when you do it, you actually start to sink this stuff into your memory banks. So that's spectacular. Let's go ahead and identify some things. Anytime here that you see, let's identify this thing, that anytime you see something like this, this is actually what the thing looks like in its beauty. Now I'm showing that this DNA is completely condensed. This is what the whole thing looks like. So if I took this part right here, and did a blow up. So I'm going to take this thing. Man, I wish I had another color to do this with. I'm going to actually just use one of my chalk colors to do it. So if we took this part right here and we did a blow up of this thing. So now we know what that thing is, and every chromosome is in this state. We call this equal to what? A, in the singular, duplicated chromosome. An A duplicated chromosome is equal to what we call 2 chromatids in a centromere. And so what I'm going to do is identify these things. 2 chromatids in a centromere. 2 chromatids in a centromere. Here's a chromatid. Here's a chromatid. And then this particular area here, I'm going to get out another color. I'm going to throw some orange structures in here. So this orange bit right here, this area right here is called our centromere. And I'm drawing some proteins here. These are called kinetochore. Proteins. Okay, so now it's time for us to look at a couple of images from the textbook that'll really help us understand things. So you remember that we've got duplicated chromosomes, and those duplicated chromosomes are going to condense and become visible. This is a duplicated chromosome right here. They're calling it a condensed chromosome. I would actually change that name to duplicated condensed chromosome, because here we see 1 chromatid, 2 chromatids called sister chromatids, there you go, and a centromere. So this is a duplicated condensed chromosome, 2 chromatids and a centromere. 2 chromatids and a centromere. Get used to it. So these are things that we are going to draw as we draw the process of mitosis, and then later in meiosis, we are going to draw duplicated condensed chromosomes as X's. You know, kind of squiggly, wavy X's. That's what we're going to do. So kinetochore proteins is what these are. So every one of these is actually something that started off as this really long functional chromosome, you know, unwound DNA that can do its thing. We'll talk about DNA doing its thing in the future. That's where the class kind of wraps up, is from gene to protein. But during this process of division, the whole thing has to happen with the chromosomes just twisting and turning on each other with proteins to become something that basically as a single, as just an uncondensed chromosome is invisible under the light microscope, other than shading of the nucleus darker than the surroundings, to the point when it's condensed to be a duplicated chromosome, it is highly visible under the microscope. So highly visible that you can count them. That goes way back to the beginning of this lecture when I talked about the N number. And determining the N number and ploidy is something that has to be done in the state of mitosis. You have to pick one of these and take a picture of them. There's one more thing I'm going to tell you about here on the board. There's one more thing I'm going to tell you about here on the computer with the karyotype. You have to do the karyotype when the chromosomes are duplicated chromosomes, condensed and visible. So I guess I should write that down here. So a duplicated chromosome, 2 chromatids, and a centromere. These are condensed and visible under the light microscope. And there's a beautiful figure in the book that will take you down that journey to see what one of these things really looks like. So figure 12.4a is where I want you to go. And that's A, by the way. A, 12.4a, and that's on page 236. By the way, this whole thing up here, the whole beautiful process of mitosis, is actually figure 12.7. So this whole thing pages, that's 2 pages, is a big one, 238 and 239. And double check, make sure you can see that. And yeah, you kind of can. I gotta just bump it up a wee bit for you. There you go. So there's that. It's a duplicated chromosome. So we've blown up one of these to see what they are. Now we can kind of get an idea. Remember, there's another thing that we should actually contemplate here. It's actually between moving on to anaphase, we have this checkpoint. And on that checkpoint, on either side of that checkpoint, then we would have kind of a red light here. And we'd have a green light here. And that green light, the key to that green light was actually connecting each one of these duplicated chromosomes at their centromeres to the spindles. When they're connected to both poles, then it moves on. So this is a very important checkpoint. I've just color-coded it so you can kind of remember that from the earlier lecture. Fantastic. Okay, our last slide I'm throwing in is a slide that is going to give us a little bit more detail. You might have heard me bleep out the word kinetochore. So now we actually have a really nice figure in your book that's going to show us what the spindle actually looks like. I think all of you at this point would know that this particular cell is in a state of anaphase because all of these duplicated chromosomes, these chubby X's, are on an equator. They're actually showing it beautifully, almost like a plane. They have a really nice rendition of the plane here. And you can see that they're all organized there, but there are these little blue egg structures here. These are the kinetochores. And I drew those and identified those up here on the big diagram. I've identified those in orange. Matter of fact, you can see them right there. If you look over there on this camera, there they are. So this is the way that it looks, and you can see that the centrosomes are forming the poles. And then just look at the word microtubule. It's quite abundant here. And everybody, let's go back one last time and take a look at brush off our concept of microtubules. Remember, it's a tubular arrangement of 2 subtypes of proteins, alpha tubulin and beta tubulin, kind of in a barber pole design. That is a microtubule, extraordinarily important in the cell, and obviously extraordinarily important in the process of mitosis and later meiosis. So that's it. This is hot stuff. At least it is for me, and I hope it is for you. One of the things I want to do is that now we know the whole life of a duplicated chromosome. So now that we know the life of a duplicated chromosome, know what it is, let's look at the life of the duplicated chromosome. So if we actually take our GS all the way down here, and then we kind of continue down here with what's going on between these processes. We have divisions between these phases. I wanted you to see the life of a duplicated chromosome. Technically, that duplicated chromosome starts here. The duplicated chromosomes form during S. They aren't condensed in this point, but they form here. So then the duplicated chromosome is obviously alive through G2, it's alive through prophase, it's alive through metaphase, and then when they split, that is the life of a duplicated chromosome. At this point, we have chromatids migrating to opposite poles. So at this point, chromatids migrate to the opposite poles. So at this point, we do not have duplicated chromosomes living. So it's this point right here that's the life of a duplicated chromosome. And then maybe I'll shade that in a cool color. Here's the life of a duplicated chromosome then. Now, the most important thing for us is to wrap our brains around the concept of ploidy. So we have to tell you that no matter how you count it, a duplicated chromosome for ploidy is equal to 1. This is very important. So in this thing here, a big star, and that star says, note, a duplicated chromosome is equal, for ploidy purposes, to 1 chromosome. So that's just this little thing that we need to throw in here for you to really know, because we want to trace the ploidy of this thing. So therefore, because of this, everything from here on is 2n, 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 and then each of these daughter ones that's forming is 2n. That is really, really, really cool. Everything is diploid. So we start with one diploid cell. In our case, we said diploid, or 2n, n equals 3. So we should have had a total of 6 chromosomes. We can't see them here because they're not condensed. Now we can see them, count them, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. But this is the transitional phase. Once we're over here, 1, 2, 3, 4, 5, 6 in each side. That's the beauty. Diploid all the way through. That is how we count it. Got to double check and make sure you can see my diploidy. And that... Nope, you can't. Okay, so here we go. Okay. So now, again, what I was just saying was diploid all the way through. And now you can actually see the notes on the bottom that shows, yes, this is diploid all the way through. Might be a little bit confusing for you to do this, but remember this rule, that basically a duplicated chromosome is not a chromosome from counting perspective, and either is a chromatid. A chromatid is not. So 2 chromatids is equal to 1 chromosome for counting purposes. 1, 2, 3, 4, 5, 6. 2 chromatids equals 1 chromosome. That's what I'm trying to say. We need to fundamentally view this process as basically diploid through to diploid. That's how we do it. These are not functional chromosomes. I think that's where my definition suits. So that's that. Okay, so that is the beauty of mitosis. What we do next is move on into that extraordinary domain of meiosis. And There's some new curveballs, new complexities, but once you understand meiosis, you're completely prepared to march into the wonderful world of inheritance. You know, this crossed with this. What gametes does it produce? What does it produce ultimately? Oh, it gets fun. So that's all we've got for today. I hope that you enjoy this, and I hope you do your jobs. You know your jobs. You've got notes to make. You've got quizzes to do. And some of you have an exam maybe to brush up on. I'm going to be posting a new exam very soon. That'll be exam 3. You have a schedule that I sent you that shows the breakdown of color-coded breakdown of lectures and exam. And man, we are on the downhill slope, but boy, it's an exciting downhill slope. We've picked one of the best slopes to ski down, and it's going to be fun.